New admission. First class.
New students, sir.
Okay. Please introduce yourself. Name. Sit down, sit down. Okay. Okay. From when you have started your preparation, means for competitive exam, last 2 months. What you have done till date? From 2 months. Okay, fine. Actually, teach science and technology. Almost the subject is over. Tomorrow will be the last class. Today we'll discuss biotechnology. Tomorrow, remaining topics, robotics, then information technology, nanotechnology will cover tomorrow. Yesterday we were discussing about in the last class different types of energy resources. Earlier we were discussing about different types of hackers, then different types of ships we have discussed, submarines we have discussed, right? In our armed forces, we have how many classes of submarines? 5 classes of submarines we have. Shishuma class, Sindhu Ghosh class, Akula 2 class, Kalvari class, then Arihant class of submarines. Submarines are basically classified into submarine ship, hunter killer, diesel electric powered. Later we have SSN, submarine ship nuclear powered. Then SSB, submarine ship ballistic nuclear, which can carry ballistic missile also. Right now we don't have any submarine of the category SSN, submarine nuclear we don't have. Yet to be commissioned, the Akula 2 class Chakra 3 submarine. Actually, the commissioning was planned to happen in 2025, but due to some various technical issues, it has been postponed to 2028, 3 years delay for the commissioning of the Akula 2 class Chakra 3 submarine. Then we have discussed about the different types of warships we have, right? We have aircraft carrier. How many aircraft carriers we have? 2. INS Vikram, INS Vikramaditya. Vikrant and Vikramaditya we have 2 aircraft carriers. Then we have destroyer ships. Destroyer means which fight, attack. Destroyer ships we have. Then we have frigates, lesser in size compared to the destroyer ship. They are generally defensive ships, right? Defensive arm of our navy. Then we have corvette, still smaller ships compared to the frigates, right? Biotechnology. Bio means life, technology. Technology dealing with life, living organisms, or simply we can say, how do you make curd at home? How do you prepare curd at home? You heat the milk. When the milk is little warm, then you will add some curd, buttermilk, etc. Right? Later, when you keep it all night, it will turn into curd. What you are adding curd itself that contains various microorganisms, that is mainly lactobacillus, which helps in the conversion, the fermentation or conversion of milk into curd, lactobacillus bacteria. With the help of this living organism, we have converted milk into curd, right? In the industry, let's say KMF, Nandini, how they make curd? Do they heat a lot and lots of milk and add some curd to it? No. They will use special chemicals, enzymes to convert milk into curd. Special enzymes they use, right? Likewise, how do you prepare bread? With the help of yeast, right? Brewer's yeast. With the help of yeast, fermentation takes place. Fermentation. In these mechanisms, we do use living organisms, microorganisms. With the help of living organisms, we are doing some work, right? Something is happening. The end product is useful to us. The major agenda or use of technology is to get a useful product for us, the humans. Okay. With the help of biotechnology, one thing, biotechnology is classified into different subfields like food biotechnology, agricultural biotechnology, medical biotechnology, marine biotechnology, etc. Biotechnology. Using of living organisms or enzymes to get the useful product. That's it. Biotechnology. Biotechnology, the development, earlier it was called as genetic engineering, the branch genetic engineering. Later the name biotechnology came. Biotechnology. During the discussion about a different type of fuels, we have discussed that the biofuel, different categories, first generation, second generation. Do you remember? First generation biofuel developed from food-based product, sugarcane waste, etc. Second generation from non-food crop. Third generation from algae microbe, etc. Fourth generation, genetically modified crops. Generations. Later, we have discussed about hydrogen. That too, gray hydrogen, brown hydrogen, blue hydrogen, green hydrogen. Do you remember? Different types of hydrogen means how the hydrogen is manufactured based on that. The color coding. Likewise, we have color coding here. Red biotechnology, green biotechnology, blue biotechnology. Likewise, red biotechnology means medical biotechnology, red biotechnology. Agricultural biotechnology called green biotechnology, agricultural biotechnology. Then industrial biotechnology, gray or white, generally gray biotechnology. Then marine biotechnology, blue biotechnology, blue biotechnology. If in case you get any bacterial disease, let's say tuberculosis, typhoid, etc., you do get a medicine by consulting the doctor, right? The doctor will suggest a course of antibiotic medicine. Antibiotic means anti means against, bio means alive. The medicine which kills the bacteria which is affecting us, right? If our immune system is stronger, we can kill that bacteria, but we couldn't kill that bacteria microorganism. That's why we got the fever disease. To kill that bacteria or microorganism, we are using antibiotic medicine. World's first antibiotic that was used during the time of First World War itself. Penicillin. Penicillin, world's first antibiotic, developed by Alexander Fleming. Alexander Fleming, developed from this injection, penicillin injection is developed from fungi, a microorganism. It is developed from Penicillium notatum fungi. Penicillium notatum. Penicillium notatum fungi. Penicillin injection developed from that Penicillium notatum fungi. It's a living organism. The medicine which we have developed antibiotic that helps to kill the microorganisms. Right. This is biotechnology. This is not the complete biotechnology. This is also biotechnology. Right. Then during the time of corona, to detect the disease, which tests we have underwent? Corona test name. Do you remember? There were two tests by taking the nose and throat swab. Right? Throat swab. Exactly. Two tests. One is rapid antigen test. The test would give immediate result. Another is RT-PCR, reverse transcriptase polymerase chain reaction test, RT-PCR. We'll discuss this in detail later. Reverse transcriptase polymerase chain reaction test. That a rapid antigen test, if the viral load is more, if the viral load is high, it would give the result immediately. If not, if you get negative in rapid antigen test, then also the doctor, the lab technician would suggest you, please, for at least 2 days, stay quarantine so that after 2 days you will get the proper report result from your RT-PCR test. To diagnose the disease, to detect the disease, we are doing many tests, right? For example, RT-PCR. This is also biotechnology. Biotechnology means by using living organisms, by using their products, by using enzymes, getting useful product or useful effect for human beings as that's it. To detect disease, to curb the disease, to cure the disease, that is biotechnology, right? To detect AIDS disease, AIDS, viral disease, bacterial disease, AIDS, viral disease from the virus HIV. Generally, we call HIV the disease, but actually the disease name is AIDS. The virus name is HIV. Acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome caused by human immunodeficiency virus. Human immunodeficiency virus, HIV. To detect this disease, we make a test called ELISA. We'll discuss these terms later. Just listen now. Later, you can write. Enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. ELISA test. Biotechnology. Right? To detect the disease. Good morning, sir. To detect the disease, diagnosis, then for treatment, etc. This is related to medical biotechnology. We have antibiotics, we have vaccines. Vaccine, how vaccine work? We all have got many number of vaccines from our childhood, right? Polio vaccine, DPT vaccine, BCG vaccine, many vaccines we have got. We didn't had any disease earlier in our childhood, right? Then also we were injected with many vaccines. Why? Precaution for the disease not to come in the future. That's why we got the vaccine, right? We got polio vaccine so that we should not be affected with polio in the future. That is the case. Then why we got corona vaccine even though many people got cured from corona disease? We got corona, we got cured. Later, then also they have administered vaccine to us, right? Reason? In the future, if in case the more stronger variant of coronavirus comes, it would be easier to fight against it. That's it. Vaccine, right? These all things comes under medical biotechnology, and one more important thing, genetic engineering, gene therapy, genetic treatment, etc., deals with the medical biotechnology. For example, there are so many genetic disorders, genetic diseases. Let's say, for example, cancer exactly cannot be treated with this biotechnology. Cancer can be treated with the help of radiation therapy, chemotherapy, etc. A disease like hemophilia. Have you heard the name? Have you heard the name anemia? Anemia is due to the deficiency of iron. If you don't consume proper amount of iron in your food, for example, green vegetables, green leaves, then you will get iron deficiency due to the deficiency of iron, and also due to the deficiency of vitamin b6. In your school days, you might have got a small iron tablet. Do you remember? That is folic acid tablet. Folic acid, chemical name of vitamin b6. Chemical name of vitamin b6. There are so many types of anemia. Majorly anemia is caused due to iron deficiency, then vitamin b6 deficiency. How many vitamins do we have? We have discussed all these things in the earlier class. Vitamin A, vitamin B complex. Under the vitamin B complex, we have many. Then we have vitamin C, D, E, K. Under D, we have categories. Let's say D. Then E and K we have, right? Vitamin A, D, E, K. A, D, E, K, fat soluble vitamins. Vitamin B and C, water soluble vitamins we have. Under vitamin 1, B, we have B1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Many vitamins we have. They're important vitamins. Vitamin B1, B2, B3, B6, very important vitamins, let's say. B6, then B12. In many times they have asked in the examination, right? Here vitamin B6 deals with B6, B9 deficiency, vitamin B9, vitamin B6, vitamin B12, different types of anemia it causes, reduction in RBC count. Okay. Hemophilia is the disease. Hemophilia, it's a genetic disorder. Who's the father of modern genetics? Who discovered heredity? Do you know what heredity is? Heredity. From generation to generation, the traits, hereditary traits transfer from father, mother, or grandparents to the next lineage, right? Heredity. Father of modern genetics, or the person who discovered heredity, is Gregor John Mendel. Gregor John Mendel. John Mendel. He discovered heredity by studying 7 years on garden pea plant. Normal peas means there are varieties of peas, chickpea, horse pea, right? Garden pea means batani. Garden pea plant he studied on. Then he discovered heredity by studying about the garden pea plant, father of modern genetics. Genetical disorder, hereditary disorder means the disease which comes from birth, from generation to generation, disease which comes from birth. For example, there are so many types of diseases which we have discussed in the earlier classes, like Turner's syndrome, Klinefelter syndrome, Down's syndrome, etc. Those are the diseases where number of chromosomes are varied. How many chromosomes do we have? Chromosomes. 3 extra. 23 pair of chromosomes. Total we have 46 chromosomes. 23 pair. We have chromosomes in pair. In female, you have XX chromosome, male XY chromosome, right? 23 pair total, 46 chromosomes we have. In the diseases like this, Downs, Turner's, Klinefelter's, etc., they will be having a varied number of chromosomes. Downs syndrome, 47 chromosome. Turner syndrome, 45 chromosome. Klinefelter syndrome, 47 chromosome, like this. Numbers are varied. A person born with these kind of genetic disorders that cannot be cured with any type of medicine, that cannot be cured even with genetic engineering. No cure. That's not possible to cure that disorder. But there are some diseases like hemophilia, thalassemia, then sickle cell anemia. Those diseases can be cured with the help of gene therapy, gene treatment. Thalassemia, hemophilia, sickle cell anemia, many other are there. Thalassemia disease, have you seen the recent program coming in Z Power or Z Canada, I think, Idea Bazi, dubbed program from Hindi. Have you heard about the name Shark Tank program? See, there is a program where entrepreneur who created a new business will comes in front of the investors. You have so much money, you are ready to invest in new ventures, new companies. Okay, I am an entrepreneur, I have an idea, but I don't have that money. I will come in front of you and I will pitch my idea. I am starting this new company, so and so company, with this goal. This is the product, this will be the cost, this will be the profit percentage, etc. If you are interested, you will invest in the company by taking some share, right? In that program, one family went there in the Shark Tank program 2 3 years back. That family went there to raise money, to earn money, to treat their child. She is suffering from thalassemia, the disease. There was a deadline. Within the age of 7, she should be treated by gene therapy. Otherwise, it is not possible to treat in the future and she will die in few years. Thalassemia disease. For gene therapy, she required how much money? 2 plus crore rupees. Just imagine. Not the normal injection medicine treatment, special treatment, right? Gene therapy. Gene therapy means what? See, where exactly the gene is present in our body? Each and everywhere. In each and every cell of our body. How many cells do we have? What is the size of the cell? Centimeter, millimeter, meter, kilometer, micrometer, nanometer. We cannot see our cell, right? It is very small. It's in the size of micrometer. Virus is in the size of nanoscale. Virus, nanoscale. Bacteria, microscale. Cell, microscale. Okay. How many cells do we think we have? Million, billion, trillion, million, 10 power 6, 10 lakh, billion, 10 power 9, 100 crore, trillion, 10 power 12, 333, 10 power 6, 10 power 9, 10 power 12. Okay. Trillion. India will become 5 trillion economy, right? 5 trillion. Trillion means 1 lakh crore, 10 power 12. Crore 7 zeros, lakh 5 zeros, 7 plus 5, 12 zeros, right? Adult human being, adult human being contains trillions of cells. Trillions of cells. Just imagine, we have trillions of cells of a size few micrometer. We have different type of cells, right? You are multicellular organism. Are we eukaryotes or prokaryotes? Eukaryote means an organism which is having a cell with definite nucleus. If nucleus is present, eukaryote. If nucleus is absent, prokaryote. We are eukaryotes. We have nucleus. Only bacteria and blue-green algae, they are prokaryotes. Only bacteria and blue-green algae, they are prokaryotes. Other than those, other than bacteria and blue-green algae, all other organisms which you can name in this world, all are eukaryotes. They contain the definite nucleus. Okay. Inside the nucleus, we have what? Inside the nucleus, we have chromosome. XX in the case of female, XY in the case of male. We have chromosomes, right? Inside the chromosome, we have what? DNA. We have DNA. Here also we have. Similarly, here also we have DNA. Inside the DNA, we have genes. Inside our body, we have trillions of cells. Inside each cell, there is a nucleus. Inside the nucleus, we have chromosome. Inside the chromosome, we have DNA, chromosome. Then inside the DNA, we have genes. Gene editing, gene therapy means replacing the faulty gene with the corrective gene. There is a gene which is having a problem that is causing this disease, thalassemia, hemophilia, etc. That should be removed. Other gene should be added. See, if this button is not proper, I can remove it and add it. The work is easy. It is not that complex. But just imagine, size of the cell is micro. Inside that chromosome, DNA gene. Very complex, right? That's why the amount 2 crore rupees. Very complex thing to do. Gene editing, gene therapy, gene treatment, right? The disease thalassemia means natural capacity to produce hemoglobin is reduced. Here, natural capacity to produce hemoglobin is reduced. Thalassemia disease, thalassemia. That means, what does hemoglobin do? What is its work? Hemoglobin. It gives blood the red color. First thing, it's a coloring pigment. It gives blood red color. In the blood, we have a red blood cell, white blood cell, then platelets. RBC, erythrocyte, WBC, leukocyte, then platelet, thrombocytes we have, right? Inside the red blood cell, we have hemoglobin. We have hemoglobin inside red blood cell. The main function of hemoglobin is, one, it gives color. Other important function, it carries oxygen. Oxygen carrier of human blood is hemoglobin. See, I need my hemoglobin to carry this much oxygen. For this, I need 100 hemoglobin, let's say. For example, if my body produces only 30 hemoglobin, then it cannot carry this much oxygen. It will carry only this much. Then what will happen? Energy will not be produced in my body. Energy will not be produced. I will feel dizzy. I will feel sober, right? If the oxygen produced or hemoglobin produced is very less, then what happens? The person might die. A person suffering from thalassemia, only treatment, only cure is gene therapy, gene treatment. Temporary solution, blood transfusion regularly. In this boy's body, thalassemia, due to thalassemia, hemoglobin is not producing a proper amount. Then he requires hemoglobin. So he need to take blood from other person regularly. every week, 15 days, 1 month, depends on the frequency, based on his requirement, based on his age. He needs blood regularly. So generally, a person suffering from thalassemia, his lifespan will be very less. Only treatment, gene therapy, thalassemia. Then we have hemophilia, also called as royal disease, because this disease is seen in the royal family, Britain's royal family. That's why the name royal disease, hemophilia. Blood does not clot. It is a disease where if you get any wound, the blood will continuously flow. It will not clot. That kind of disease, hemophilia. Natural capacity to produce clot will not happen here. Blood will not clot. Hemophilia. Rare genetic disorder. These are all the rare diseases. Rare genetic disorder. Hemophilia. If a person having hemophilia, what will happen? If any accident, etc., happen, blood will continuously flow out. Due to blood loss, the person might die. Only solution, he should carry the coagulant medicine with him in case of emergency. Like we do use turmeric, right? Turmeric for faster clotting. Likewise, there are many over-the-counter medicines available. Medicines, with the help of that, the blood can be clot easily in a faster rate, right? Then we have sickle cell anemia. What is the shape of a red blood cell? That means round, circular. From the front, if we see, it is like this only, as circular. But from the side, it is like this. See, thick crust pizza. The edge will be bulged, inner part will be hollow curved inwards, right? That kind of shape. This shape is called as biconcave shape. The shape of red blood cell, biconcave. Concave means the shape of the lens curved inwards both the sides, right? Likewise, biconcave shape of our red blood cell. Sickle cell anemia means due to abnormal hemoglobin, shape of a red blood cell turns to sickle shape. Sickle means sickle. ಕುಡುಗೋಲು 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 ಕೇಳಿದಿರಾ? ಕುಡ್ಲು ಅಂತಾರಲ್ಲ ಚಿಕ್ಕದು. This kind of agricultural equipment, right? The biconcave shaped red blood cell is converted into sickle shape. This shape, half moon, curved shape. This red blood cells will be non-functional. They will not help the transportation of gases or anything. So, see, in our blood, the majority of the cells are red blood cells. The majority of the cells are BC red blood cell. If the count decreases, anemia. Normally, anemia happens due to the deficiency of iron or vitamin B6, B9, right? But here, what is happening? Due to the deficiency of hemoglobin, RBC actually, because due to the abnormal hemoglobin, RBC count is reduced, red blood cell count reduced. The shape of the red blood cell converted from biconcave to sickle shape. Inactive, non-functional sickle shaped, that is sickle cell anemia. Normal anemia can be treated by giving you vitamin B9 folic acid tablet, vitamin B6, pyridoxine, vitamin B12, cyanocobalamin, or iron tablets. With the help of medicine, that can be cured normal anemia, but this anemia cannot be cured with the help of medicine. They also require a regular blood transfusion. A regular blood transfusion, they also require. Sickle cell anemia. These kinds of diseases can be cured with the help of what? Gene therapy. Then we have agricultural biotechnology, green biotechnology. Agricultural biotechnology, let's say, see in the earlier days, what was the shape of or size of guava? Seaburn. It was very small. The seeds inside it was very hard. Right? Now the shape size of the guava very big, nearly 300, 400 grams, 500 grams also. Right? It's soft, more tastier compared to the earlier ones. It depends from breed to breed. Then papaya. Earlier papaya used to get ripen or get soft very fast. Today the color turns yellow orange. Tomorrow it will be soft, fully ripen, difficult to cut, peel itself. But right now, many days we can keep. It is completely orange. Earlier, have you ever seen a papaya that is completely orange? Very rare, right? Right now, the color is fully orange. Then inside, very less number of seeds or no seeds. Then more flesh means the fruit which we can have. Then it is hard. It will become soft very slowly, right? Hybrid varieties. These hybrid varieties of fruits, vegetables we can develop by biotechnology. Biotechnology. Then in India, Green Revolution started under the leadership of, in the world, Green Revolution started under the leadership vision of Norman Borlaug in the world, in America. In India, under the leadership of, recently, a year back, he died, M.S. Swaminathan. M.S. Swaminathan, father of Indian Green Revolution. What is Green Revolution mean? Green Revolution? Increase in production of food crops. Increase in production. Which crop actually? Exactly. Which crops production was increased during the time of Green Revolution? Paddy and wheat, rice and wheat, Godi, right? Where exactly Green Revolution happened in India? In Punjab, Haryana, Western Uttar Pradesh, that region. There. How Green Revolution happened? He, M.S. Swaminathan, under his vision, based on the inputs from the earlier Green Revolution which happened in the world, he developed a HYV seeds, a special seeds. HYV means high yielding variety. High yielding variety seeds. High yielding variety seeds. And he started using many fertilizers, many insecticide, herbicide. Herbicide means, herb means small plants. Weedicide we can say, weed, kalenashaka, that kind of medicines he started using. With the help of that, he got a good production, huge amount of production compared to the earlier one. Then nutrition enrichment with the help of biotechnology we'll discuss later. Industrial or gray biotechnology for the industrial scale production. Industrial scale production we'll discuss. Then marine biotechnology, increasing or benefiting marine life by the help of biotechnology. Medical biotechnology, use of living cells to develop technologies to improve our health and to get required amount of nutrition. A recombinant DNA technology. We'll discuss this in detail in the future. Recombinant DNA technology. With the help of this technology, it it is possible to create, it is possible to mass produce many various drugs. For example, let's say penicillin. Penicillin is developed with the help of fungi. Penicillium notatum fungi. It's a antibiotic medicine, right? Likewise, vaccines and antibiotics. Have you heard the name antigen, antibody, antibiotic, then vaccine? These are all the different terms. Antigen means which will stimulate our immune system. Which cells of our body are called as soldiers immune body? Which cell of our blood will help to fight against the diseases? Immunity, fighting against the diseases. White blood cell, WBC, right? White blood cells are called as soldiers of human body because they help us fight against many microbial diseases, many kind of diseases, right? Antigen will our immune system, stimulate our immune system. If someone slap you, it will slap him back, right? If someone pinches you, will pinch him back. Bites you, bite him back. For a reaction, there will be suitable response. Likewise, a typhoid bacteria affecting me, Salmonella typhi, suitable response should be there, right? The response should be of the response for coronavirus. It should be for the Salmonella typhi bacteria, right? Suitable response should be there. That suitable response we get with the help of this antigen. It will stimulate our immune system so that suitable response is generated. That a response is called as antibody. Antibodies are produced in our body. Antibody, immune response in our body. Antibody. Then antibiotic means, anti means against, bio means life, against life. Antibiotics are the medicines to kill generally the bacteria. Antibiotics are medicines used against the bacteria. Antibiotic, medicine. Antibiotic. See, generally doctors suggest you to take the full course of antibiotic medicine, right? Why? If you don't take the full course, what might happen? The disease causing bacteria can develop immunity against that medicine. You are giving the medicine to kill the bacteria, right? If the medicine is not to the fullest effect, what will happen? That bacteria will develop immunity against this medicine. For example, few years back, during my phase of UPSC preparation, this question was very much in news. Multi-drug resistant tuberculosis bacteria. Multi-drug resistant tuberculosis bacteria. Tuberculosis disease, you know, right? Which affects our lungs. Tuberculosis, TB, caused by Mycobacterium tuberculosis bacteria. Mycobacterium tuberculosis bacteria. This tuberculosis disease got immunity against many medicines which was available in the market. During that time, no medicine was able to cure the disease. The main problem, the main culprit are the doctors. They have prescribed many unwanted number of antibiotics. That's why the bacteria developed immunity against it. Later, awareness was created. A special new medicine was developed. Right now, restricted use is there. If that also not maintained properly, then it will develop the resistance again. Right? That is the case. Vaccines. Vaccine means precaution measure, right? Basically. See, once upon a time, in the earlier days, half of the Europe died due to a disease called smallpox. Have you heard about the disease smallpox? Amma, sidubu antaralva, skin rashes kind of thing, big moles which happen. That will that was very normal in India also in recent years. If a person gets smallpox once in his lifetime, it will not be repeated again. That was the thing you might have heard, right? If you get smallpox once, it will not come again. Rarely it comes again to few people, right? Smallpox. Is smallpox fatal in India? Fatal means will death happen? No. What medicine you would have taken? No medicine, just rest. Then basic Ayurveda or the medicine which you have heard from your elders, etc., like bathing with water mixed with neem, then applying various sandalwood, etc. Not exactly the sandalwood, that horn of some animal, etc. Right? It's the deer, I think, but that will not help. Obviously it will go on by its own. If you do that also or not also, it will go within few days. That's the case. With the help of our immunity, that will go. Here, during that time, the smallpox, was it a viral disease or bacterial disease? Smallpox, viral disease. Variola vaccine is the name. Variola family virus. Wait, the smallpox that was caused killed many people, right? In the Europe, there was a person called Edward Jenner. Edward Jenner. He observed many people are dying from smallpox. There was a similar disease called cowpox. Smallpox, which was affecting humans majorly, cowpox affecting cattle, cow, sheep, buffalo, etc. Cowpox. There were few women who their life means their living is based on selling milk. Okay, the milkmaids, they used to get the cowpox disease. Once they get cowpox disease and gets cured, they will not be getting the smallpox disease. Cowpox, it was not fatal. The disease effect is very less. So he thought, if you get smallpox, if a person gets smallpox, he might die. If a person gets a cowpox, it will be cured within few days, right? It is better to get cowpox, right? If a person get cowpox, he will not be getting smallpox again. That's what he did. He took the virus which is causing cowpox named variola vaccine. Variola vaccine. From that name, the vaccine, the name came. World's first vaccine developed by whom? Edward Jenner. World's first vaccine developed by Edward Jenner. He developed world's first vaccine, variola vaccine. Variola vaccine. He developed the vaccine for which disease? Vaccine for smallpox. He developed. See, during the time of corona disease, we all have got the vaccine, right? Covishield, Covaxin. After giving you the vaccine injection, they might have given you $1 tablet, right? Why? Gift along with the vaccine. Hmm? Why the tablet? All vaccines are weak or dead microorganisms. All vaccines are weak or dead microorganisms. We are taking in the dead, weak coronavirus. That will also be having some effect on us, right? Mild disease might come. If you get mild disease, to treat that, the dolo. In our childhood also, after getting vaccine, many kids do get fever for few days. That's normal. That is the effect of vaccine which is going on there. In few people, effect of vaccine will be subtle, normal, without any fever. In few, reaction will be same, fever, right? All vaccines are weak or dead microorganism, or their genetic material, or their protein structure, right? Why? See, you do attend many mock examinations, right? Before attending the original examination, you do attend a mock examination. Why? To know the pattern of examination, to know the syllabus, to know how the examination will be, to feel that. If the mock test itself is very difficult, you will be scared to write even the mains examination, proper examination, right? The mock test should be little easy so that you will get that confidence to fight, to attend the original examination. Likewise, the vaccines will be dead or weak bacteria. By fighting against that dead or weak bacteria, you will get immunity. So next time you will not get the disease. If in case you get the disease, you can easily fight it back. That's the case. The polio vaccine which we have taken till the age of 5, that is also weak or dead polio virus, right? Polio vaccine developed by, discovered by Jonas Salk. Polio vaccine, Jonas Salk. Vaccines are weak or dead microbes, weak or dead microorganisms. Which are weak or dead microorganisms. Example of medical biotechnology. Genetically engineered insulin. Have you heard the name insulin? Insulin is naturally secreted in our body. Where exactly in our body? Pancreas. Okay, in the pancreas gland. Adrenal gland secretes adrenaline, emergency hormone. That is during the time of extreme fear, etc. In the time that is secreted, adrenaline is present above kidneys, above kidney cap-like structure. Pancreas below the stomach, little behind pancreas. In the pancreas, insulin hormone is secreted. What is the function of insulin? When generally a person takes insulin injection, when he has diabetes, sugar, right? Sugar diabetes means what? In the blood, the amount of sugar is more. Which sugar is present in our blood? Sugar name. Glucose. Blood sugar is glucose. If the amount of glucose is more, then insulin will control that. Blood sugar level is controlled by a hormone called Insulin, blood sugar level. If insulin cannot control the level of glucose present in our blood, that condition is called as diabetes. During diabetes, the insulin cannot control the amount of sugar. That's why we have more amount of sugar in our blood. Either insulin is secreted less or we are consuming more sugar. That is the case, right? Genetically engineered insulin producing insulin or helping the pancreas secrete more insulin. Production of insulin in the laboratory. See, the insulin injection which we are taking is not extracted from any human being, right? It is developed in the laboratory itself by using many chemicals. That is genetic engineering. Production of insulin in the laboratory. Insulin is a enzyme. Insulin, it's a hormone. Hormone. Hormones are generally called as chemical messengers of human body. Chemical messenger. Why? It will maintain something. Here, insulin is maintaining our blood sugar. It is maintaining something. Thyroxine hormone, it maintains rate of calorie burnt. Likewise, it does some job. Then gene therapy. Therapy means treatment. If a person is born with a hereditary disease, name any hereditary disease we have discussed today. Hereditary disease. Sickle cell anemia, hemophilia, thalassemia, etc. These are genetic disorders, right? Many are there. These are few important genetic disorders. If a person is born with a hereditary disease, genetic disease, is it possible to cure it with the help of any medicine? No, it is not at all possible. But that can be corrected. That can be corrected with the help of gene therapy. What does gene therapy means? Removing the faulty gene, adding the corrective proper gene. That's it. Gene therapy. Genes are inserted into the person's cells and tissues to treat that disease. Correction of gene, removing the faulty gene and adding the normal gene. That's it. Gene therapy. This can be done in various different methods. Gene therapy, it's a procedure, right? It can be done in various methods. We'll see later. With the help of gene therapy, what can be done? Hereditary diseases can be cured. Faulty genes can be corrected, right? There is an example. Curing adenosine deaminase deficiency. Adenosine deaminase. Adenosine deaminase. It is an enzyme. See, we do know, speak about many hormones, enzymes which are famous, popular. Instead of those, we have many n number of enzymes, chemicals which are secreted in our body, right? For example, let's say sex hormone of male, testosterone, female progesterone and estrogen, right? Other than that also, there are so many hormones present in both male and female. Likewise, there is an important enzyme called adenosine deaminase that should be secreted normally, naturally in each and every one of us. If it is not secreted properly, our immunity will be less. Immunity will be less. That is the problem. Crucial for immune system functioning. If a person is having this disease, what will happen? Less immunity. He will get disease very often. Smaller, weaker microorganisms can also affect that person. Immunity will be less. He cannot fight back against the disease causing microorganisms. This can be treated with the help of gene therapy. Through gene therapy, what can be done? See, listen. Which blood cell is related to immune response? Soldiers of human body. Hmm? White blood cells, right? Immunity means white blood cell. White blood cells are called as leukocytes. Leukocytes. See, erythro means red. Cytes means cell. Branch of biology which deals with the cells called as cytology. Cyte means cell. Erythro red cytes cells. Likewise, cyte means cell. Leuco means colorless. If we call that cell colorless, it will feel bad. That's why we are calling it as white blood cell. White blood cells don't have any coloring pigment. They don't have any coloring pigment. They don't have hemoglobin. That's why the name leukocyte. That's why we call it as generally as white blood cell. White blood cell's main function is immune response, fighting against the bacteria, virus, other harmful microorganisms. That's it. Lymphocytes, white blood cell from the blood of the patient. Patient means the person affected by adenosine diamines deficiency. It is taken. Adenosine diamines deficiency. It is taken. Then, what happens? Functional means active, good, normal gene from a other person. From the other person, it is taken. It is added to a carrier DNA which can carry the new normal adenosine deaminase gene. Okay. With the help of this carrier DNA, right now this person is having adenosine deaminase deficiency. We have collected white blood cell from him. Blood is extracted, later white blood cells are separated. That white blood cells we have in the laboratory condition. Right now what we are doing, we have taken blood from other normal healthy person. We have separated the gene which is having adenosine deaminase. We have attached it to the carrier DNA. That gene is infused into this blood sample. After some days, what will happen? All the blood cells present here will be having the normal gene. All the blood cells here will be having the normal gene. Then this blood sample is given to the affected person, patient. Later, only some sample is given, right? Not the complete blood is cured. Later, after some days, the older cells will die, new cells will form. By that time, the new cells formed will be having adenosine deaminase enzyme. The disease is cured right now. Okay. Gene therapy means it is practically not possible to take out all the cells and treat all the cells and give it back. Not possible. They will take the sample. If, for example, deficiency or problem is in white blood cell, lymphocytes are taken out. To treat sickle cell anemia, what should be taken out? Red blood cell. Likewise, those sample will be taken out, that will be treated, and it will be infused back into the person. That is the case. Adenosine deaminase related to immunity. Medical biotechnology to treat the disease, to detect the disease, diagnosis. Molecular diagnosis means to treat the disease effectively, we should understand what is the causative microorganism, right? Which microorganism is causing the disease, we should know. Then we should know how much amount of microorganisms are present to treat it better. The dosage, we should decide, right? For that molecular diagnosis, there are so many tests to detect the viral load, bacterial load in the microorganism. First is recombinant DNA technology. Recombining the DNA to detect the number of virus, etc., present in the body, recombinant DNA technology. Then PCR or RT-PCR. RT means reverse transcriptase. Reverse transcriptase polymerase chain reaction test, RT-PCR. Reverse transcriptase polymerase chain reaction test, RT-PCR. To detect the coronavirus. Then ELISA, enzyme linked immunosorbent assay to detect HIV virus, AIDS disease, HIV virus. See, molecular diagnosis means very less number of low concentration of microorganisms are present in your body. How much time coronavirus take to develop and affect your body? Few days, 5, 6, 7, 8, 9, 10 days. Till that it will be in kind of resting phase. Incubation phase it will be in. Okay. The amount of today you interacted with a person affected with coronavirus, you got the disease. After how many days you will see the symptoms? 3, 4, 5, 6 days. At least 3, 4, 5 days. Right. Till that time, you will be having the disease, but the number of viruses inside your body will be very less. If the disease is detected in the earlier phase, it is easier to treat it, right? If the number of viruses are less, easy to treat. That's why these kind of tests are made. Molecular diagnosis. Lower concentration, very lower concentration of microorganisms can be detected with the help of these kind of tests by amplification, by increasing their concentration. RT-PCR, ELISA, etc. are the examples. HIV can be detected by RT-PCR, reverse transcriptase polymerase chain reaction test. The main test to detect is ELISA, enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. Even with the help of RT-PCR, we can detect a cancer also. Actually, to detect a cancer, we do which test? Cancer detection test. A small amount of tissue is taken. Tissue, blood, bone, etc. Various types of cancers are there, right? Test name? Even I forgot. Will tell you. ELISA, enzyme linked immunosorbent assay. ELISA test works on the principle of antigen antibody interaction. Antigen, it will stimulate antibodies produced in the body. Antibody immune response. Based on this, the disease can be detected. If particular antigens are present or antibodies are developed, by detecting them, the disease can be confirmed. If this antigen is there, means HIV is there. If these antibodies are there, means HIV is there. That kind of test. Okay, not exactly checking the virus, checking the reaction which is happening inside the body. The earlier stage of detection can be done. Very important technology in gene therapy. CRISPR, clustered, regularly interspaced short palindromic repeats. Clustered, regularly interspaced short palindromic repeats. CRISPR technology. Cancer test name, biopsy. Clustered regularly interspaced short palindromic repeats technology. Very important, powerful method of editing the genomes, the genetic structure. We can easily alter the DNA sequence with the help of this technology. CRISPR-Cas9. Cas9 is the protein. Cas9 is the protein gene editing tool. This CRISPR-Cas9 is called as molecular scissor. Scissor to cut. Practically, you cannot literally cut with the help of the scissor. It is just an indicative name. Molecular level, it will remove the defective gene and it will help to add the proper original normal gene. That is it. CRISPR-Cas9 gene editing tool. Have you heard about a fingerprint? We have fingerprint. Our fingerprint is unique, right? From me, your fingerprint is unique. That's why under the biometric traits, we do consider our fingerprint, our facial aspects, our iris. In other, they do scan all these things, right? Our iris scan, they take face, they take fingerprint, they take fingerprint, unique. But this fingerprint can be modified. Is it possible to modify the fingerprint which we have with any operation, etc.? That can be done, actually. It is not done in the first case, but that can be. At least it can be altered. It can be removed, right? But this cannot be altered at any case. What? Our DNA fingerprint. Our DNA fingerprint cannot be changed, altered. Like the normal fingerprint, DNA fingerprint is unique. That's why the name DNA fingerprint. There are no fingers in DNA. The name fingerprinting means as our fingerprint is unique, DNA sequence is also unique. That's it. DNA sequence which is unique. That's why the name DNA fingerprint. We have discussed, one second. How DNA and RNA, the shape, etc. Right? These are called as nucleic acids, RNA and DNA. DNA, deoxyribonucleic acid, RNA, ribonucleic acid, DNA, RNA, right? See, this is our DNA. Double-stranded structure or helical structure, just like the twisted ladder, double-stranded structure. This double-stranded structure of DNA was discovered by Watson and Crick. Watson and Crick model, we do say, right? This structure, DNA. Then ribonucleic acid, RNA, deoxyribonucleic acid, DNA. The sugar which we have here is ribose sugar. Ribose. In DNA, we have deoxyribose sugar. Deoxyribose sugar. And very important thing, see, this is the backbone. This backbone consists of sugar phosphate bond. Sugar phosphate bond. Which sugar? Ribose sugar in RNA, deoxyribose sugar in DNA. Sugar phosphate bond. Then we have, see, different colors. Nitrogenous bases. Nitrogenous bases we have. In RNA, we have AUGC. AUGC we have in RNA. That means adenine, A, uracil, U, guanine, G, cytosine, C, AUGC, RNA. In DNA, we have ATGC, adenine, thymine, guanine, cytosine. In DNA, we have ATGC, thymine in DNA, uracil in RNA. Very important. And In between this nitrogenous base pairs, we have hydrogen bond. Weak bond, hydrogen bond we have between these nitrogenous base pairs. See here, yellow red, blue green, yellow red. There is some pattern, right? There is some sequence of arrangement, right? This sequence will be different from person to person. That is DNA sequencing or DNA fingerprint. That pattern will be having some similarity with our parents, not exact copy, similarity. So the lineage, the paternity, maternity can be identified with the help of DNA sample. For example, have you seen the movie Nanaku Premata? In that movie, with the help of hair strand, the daughter and mother's relationship was confirmed, right? Likewise, actually, DNA fingerprinting is majorly done to detect whether the child is their own, first thing. Then to detect the criminals, then to detect dead bodies, accident. If the body is almost or completely burnt from that ash, if you get a single cell sample, then it is possible to detect the lineage. Recently, a bus traveling to Hyderabad met with an accident. Many people died, right? In the burnt bodies, it will be difficult to detect who the person is. For the family to detect the dead person, DNA fingerprinting can be done. DNA fingerprinting. If the body is completely burnt, even not the bones are properly available, then also with that burnt ash bones, DNA fingerprinting can be done. With the help of your nail, hair, skin sample, DNA fingerprinting can be done. Any body cell sample is fine. To do this sample test, DNA sequence is very unique but cannot be altered with surgery. Our fingerprint can be altered with surgery. Fingerprint can be altered with surgery, not the DNA fingerprinting. That is not at all possible. Sir, we can do gene therapy, we can change the gene, faulty gene with the original gene, right? Will that change? Will that change our genetic sequence? Means no. That will not change our genetic sequence. The normal gene therapy, gene editing, which we have right now, will not change our genetic sequence. Generally, why do people get genetic diseases? What is the main reason? Let's say, I'll give an example. Have you heard about rare blood groups? Rare blood group. For example, Bombay blood group. Bombay blood group. The name Bombay blood group because it was discovered in Bombay, right? It is also called as HH blood group. The common antigen present in all of our blood is not available in that blood. What is your blood group? B positive. Yours? Okay, mine is O positive. You have B antigen, RH antigen. I have only RH antigen. O means nothing, no antigen, no A, no B is present, right? The dominant, prominent blood groups which we know are A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative. That's it. 8 blood groups, 4 plus 4. Other than this, there are so many rare blood groups seen in only few people. Actually, the Bombay blood group, the Bombay blood group, it is seen in one person among 4 million people around the world. 1 in 4 million around the world. That means among 40 lakh people in America, one will be having Bombay blood group. But in India, it is seen in every 40,000, 50,000 people, one people is having that Bombay blood group. Why more number of people is having Bombay blood group? Reason? In South Asian countries, Compared to the Western countries, in South Asian countries, genetic diseases are more, rare blood groups are more. Reason, close community marriages. Close community marriage, cross cousin, atta mava, maglu, akan maglu, etc. This kind of close community marriages leads to genetic diseases. See, not each and every person get genetic disease just by marrying in the close community, but if this happens for a longer duration, affect it will affect the genetic pattern. Then the person might get the genetic disorder. The basic diseases like hearing disability, vision disability, etc., and the more complex disabilities like Turner syndrome, Down syndrome, etc. Mental disability, this kind of things will happen. That's why the issue is there more in the South Asian countries. DNA fingerprinting used in forensic laboratories to identify the criminals. A criminal barged into a jewelry shop. He have stolen all the jewelry. He left no sample except his single hair strand. With that single hair strand, the culprit can be detected, right? But the culprit should be, we should get that person and test to match the sample, right? Just by the strand, we cannot go and search who that person is, but we can match, right? Forensic laboratory. Mainly the murder, crimes, etc., can be identified, detected with the help of this. Then determination of paternity, who father is, majorly. Then identifying the dead bodies. Dead bodies can be done with the help of DNA fingerprinting. Then we can study the evolution. A recombinant DNA technology. Recombinant DNA technology means this DNA sequence is not generally available. It can only be generated in the laboratory conditions. Naturally not available, generated artificially. That kind of technology. For example, mother's milk contains a special nutrient which is absent in cow's milk. Okay. In the first case, the protein, mineral, etc. present in the cow's milk will be heavier for the child to have in the few days prenatal, few days after birth. Later, after 6 months, 1 year, the child can have cow's milk. Below that, it is not suggested, right? See, a child who is not getting suitable amount of mother's milk due to various reasons, maybe death of the mother or other reason, that protein is not available in cow's milk. The child requires that protein. For that, they have developed with the help of this technology a special cow, normal cow with special ability. Cow looks normal, no physical changes. The change is that the cow milk contains that special nutrient. And one more thing, tomato. Earlier, do you remember we used to have weekly once market at our places? Weekly once market. We used to purchase all the vegetables required for our home in the weekly market because the price would be less. We would get better vegetables, fruits. The tomato, how we would select few red ripen, few oranges, so that will be ripened by the end of that week. But now that is not the case. the tomato which will be fully red, that will stay for another 15-20 days. That will not be easily softened, ripened when compared to the earlier tomatoes, right? Earlier tomatoes, what would happen? It would ripen at a faster rate. It would be softened. It would be decomposed in the faster rate. Right now, it has changed, right? Recombinant DNA technology. We'll see. Recombinant DNA technology. DNA molecules are formed by laboratory methods. Laboratory methods. Creating a sequence that would not be found otherwise naturally. It is not possible for the cow milk to have that special nutrient that can be formed with the help of this technology, recombinant DNA technology. Recombinant DNA technology, creating new gene that can not be naturally found at all. Naturally, that cannot be found, that can be produced in the laboratory conditions. That is recombinant DNA technology. From one organism, we can share the trait to other organism. That is possible here. They will be having normal phenotype. Phenotype means physical appearance will be the same. The cow will look just like the cow itself. No changes. We cannot identify physically, but that will be having the ability to produce a milk, which will be having, humans milk nutrient, right? Let's see the application of a recombinant DNA technology, widely used in medicine and research to develop various kinds of medicines, used to produce human insulin, insulin which people are injecting, that is laboratory generated, right? That is with the help of recombinant DNA technology. Then human growth hormone. Which gland secretes growth hormone? Growth hormone. We have so many endocrine glands which secrete hormones. We have pituitary pineal hypothalamus in our brain. Pituitary pineal hypothalamus. We have thyroid parathyroid in our neck portion. We have thymus gland in our chest cavity. Thymus. Two adrenal gland above our kidneys. Pancreas. Actually, it is a mixed gland. During the time of digestion, it acts as an exocrine gland. During the time of insulin production, acts as a endocrine gland. Okay. Then in the male we have testis, female ovary. These are all the endocrine glands which secrete hormone. Growth hormone is secreted by pituitary gland. Pituitary gland, master control gland, we call it as pituitary gland, right? That can be produced with the help of this recombinant DNA technology, growth hormone. Then blood clotting factor that can be developed. A person suffering from hemophilia, that can be treated with the help of this recombinant DNA technology. Then hepatitis B vaccine, etc. See, if in case there is a question regarding this technology, they will definitely give a statement explaining what this technology mean. What this technology mean? Recombinant DNA technology. Naturally, in this organism, this gene cannot be present. This gene from other organism is added to this organism in the laboratory condition, or this gene can be developed in the laboratory or taken from other organism that is added to this organism. The gene which is naturally not present is added, recombining the gene. Recombinant DNA technology, right? Very much important. CRISPR-Cas9 molecular scissor. CRISPR-Cas9. What it will do? Gene therapy. Gene therapy means removing the faulty gene, adding corrective, normal gene, right? With the help of this technology, CRISPR clustered regularly interspaced short palindromic repeat. Palindrome means from the right to left, left to right. If you read, it will be the same. For example, Navina, from left to right, right to left, same, right? That is a palindrome. That kind of palindromic repeat gene will be added here. That's why the name. Clustered, clustered, regularly interspaced here after some gap, like this, it is added. Clustered, regularly interspaced, short, palindromic repeat. Palindrome means from left to right to left, it is the same. That's it. Like that. The corrective gene is added. Cas9 is the protein. Cas9 protein. CRISPR associated protein 9. Cas9 we call it as it's an enzyme gene editing tool. See here. This is the normal gene, the upper one, normal gene present inside the DNA. Okay. This part, the target sequence, it is removed with the help of the protein called Cas9. That's why it is called as molecular scissor. Molecular scissor. Cas9 protein is called as molecular scissor. The required targeted sequence will be deleted. Then corrective gene will be added there. Corrective gene will be added. This guide RNA will help transferring the corrective gene to that place. CRISPR Cas9 nucleic acids we have discussed RNA DNA. DNA heredity related to DNA. Transgenic animal transfer of genes from one organism to other organism to create a special organism with different genes. That's it. A foreign gene is added to this organism, this transgenic animal. See, cloning is different, a transgenic animal is different. Cloning means what? Creating same ditto exact copy of the DNA sequence. Creating a human who don't just look like me, who is having my genetic sequence only, not the visual appearance. Genetic sequence, same genetic sequence. That is cloning. Similar copy, cloning. Okay. Transgenic animal means a foreign gene is added to the organism to get a required result, output. Maybe it may be the product. For example, here we do get milk. Transgenic animal, majority of the transgenic animals are mice, small rat, rats, rabbits, pig, sheep, cow, etc. There are so many transgenic animals. See, the major reason of creating transgenic animal is to detect, test the disease causing microorganism or to test the medicines, test the vaccines. See, for example, if you test a medicine or vaccine on a human, it takes more time for you to get the result. If you test the same on the mice, you will get the result in shorter duration of time, less time. It will be better, right, for the pharmaceutical companies to test. That's why they use mice, small mice. Not the normal mice, transgenic mice. Transgenic means, see, let's say, for example, They need to test corona vaccine. They need to test corona vaccine. What they will do, they will take the genes present in the human body and they will add it to the mice. Then they will test. Now the mice is having human genes, genetic structure, right? When the mice gets corona, how it will affect the mice? When it is given with the vaccine, how it will react? That will be tested. That is transgenic organism. Here, see, DNA, RNA, etc., taken from other organisms added to the normal cells of the organism. Here, eosinophil cell type of WBC, eosinophil type of white blood cell, primary fibroblast type of blood cell. Okay, it is added, the foreign gene, it is added. Later, the organism will be having, that will be injected to the organism. Then the organism will be transgenic, that will be having a different gene which it didn't have earlier, right? The first transgenic cow, Rosie, the first cloned sheep, Dolly, Dolly the sheep, first cloned sheep. See, by introducing genes from other species, other species, biological effects, etc., can be studied. For example, how the disease will affect the organism, how the medicine will act, etc. And the diseases can also be studied, how the disease will act, etc. For example, rheumatoid arthritis, cystic fibrosis, Alzheimer's, etc. on small organisms. The first cloned, the first transgenic cow, name Rosie, Rosie the cow. See, in 1997, first transgenic cow named Rosie was developed, which can produce milk with human protein, the protein which was present in the human milk, mother's milk, alpha lactalbumin. Generally, this was produced in only mother's milk, but right now it is also, actually, we do have many cows around us in the dairy market. Those cows right now are not producing this kind of milk, alpha lactalbumin, because they are not transgenic cows. They are hybrid varieties of cows, yes, but they are not transgenic. Only transgenic cows, they will produce this kind of milk if that kind of gene is used. Alpha lactalbumin, alpha lactalbumin. But main problem, there will be many side effects. There will be many side effects like allergic reaction, etc. That might be seen, that is the case. Here, one more thing, transgenic animal, transgenic organism to treat emphysema. Have you heard about the name emphysema? In the film theater, before the movie starts, a person who smokes lots and lots of cigarette, Mukesh, etc. Listen properly, they will use this term emphysema. He might get emphysema. Emphysema is a lung disease, breathing inability, emphysema. Here, it's a lung condition where you cannot breathe. The people suffering from emphysema, their air sacs alveoli in the lungs, alveoli, air sacs in the lungs will be damaged. Damaged. The treatment for this disease, that is polyketonuria, that will be the inborn error. And this emphysema, etc., that can be treated with the help of, actually directly emphysema cannot be treated. The medicine can be developed by testing on mice and small other organisms. Polyketonuria is also a similar kind of disease, natural metabolism disorder. And cystic fibrosis. Cystic fibrosis is a disease where in your will become hard, rough, which you cannot breathe through. Your mucus will become rough, scum. So breathing disability will happen. That kind of condition. To treat all these diseases, the medicine can be developed with the help of transgenic animals. With the help of transgenic animals. If you test the same medicine on human, it will take lots and lots of time to get the result. But with the help of mice, you can get the result in short span. That is the case. Transgenic animals. And to test the vaccine safety also, we have tested vaccines in the first phase on the mice, later on the chimpanzee, etc., later on the higher organisms, later on the human. Later we have used this vaccine with the help of then chemical safety testing. See, simply, we humans are selfish animals. We don't want ourselves to be tested, ourselves to be in the danger. That's why we test the medicine, vaccine, etc. on other organisms. That's it. There is a place, nuclear reactor, from where nuclear radiation might be coming. How that will affect the mice, they will test first instead of human. A chemical weapon, if it is used, what will be the effect? They will test on the mice first. Likewise, they are protecting the humans. They are doing it on other organisms with the help of transgenic animals. Transgenic means naturally that gene is not present, that is taken from other organism for generally for testing and other purposes. That's it. Take a break. We'll continue afterwards. 5 minutes. ಮಾಡ್ತೇನೆ ಬಿಡಿ pause. Shall we continue? Biotechnology in India means which are all the departments we have and what are all the programs which we are doing to help the growth in the field of biotechnology for the new companies, for the new technology, etc. What the help which we are giving. Biotechnology, Department of Biotechnology we have comes under Ministry of Science and Technology. Department of Biotechnology works under Ministry of Science and Technology. To provide services to research infrastructure, etc., to help them. Then, very important thing, implementation of biosafety guidelines. Biosafety guidelines means genetically modified crop. For example, BT brinjal. It was banned, right? BT brinjal. Do you remember? Do you know? Have you heard the name BT brinjal? In many movies, huh? BT badnakai. In one Kannada movie also it is there. Rangana Raghu steps on that brinjal. It was, I think, Rangana Raghu or Komal, I forgot. He tells Raita Virodi BT badnakai down down. Later it will blast. BT brinjal. Have you heard the name BT cotton? BT mustard? No? Okay. No worries. Mustard, saswe, cotton, brinjal, BT. Here, BT does not mean biotechnology. BT is the name of a bacteria. BT is the name of a bacteria. Here, BT means Bacillus thuringiensis. Bacillus thuringiensis bacteria. This special variety of brinjal, cotton, mustard plants are developed with biotechnology, hybrid variety. What they're doing, see, you might have seen the normal brinjal, it will get many pest attack, right? Small worms which eat the leaves of the brinjal plant and even the brinjal also. That can be protected with the help of pesticide, chemical spraying, which generally the farmers do. This method to avoid the use of pesticide to create a pest resistant plant. What? Pest resistant plant. Plants which can resist the pests, the insects. How? This Bacillus thuringiensis is a soil bacteria. It's a soil bacteria which present in the soil. This Bacillus thuringiensis bacteria produces a chemical which is naturally pest resistant. This bacteria produces a chemical that is pest resistant. Now what they have done, they have taken the gene from this bacteria, added that gene to the brinjal, cotton, mustard plant, transferring a gene, transgenic, right? Now the brinjal plant, cotton plant, mustard plant will be having a special foreign gene. Gene from which bacteria? BT, Bacillus thuringiensis. What it will do now? The plant will produce the chemical which that bacteria was producing. The plant is secreting that chemical. Now it will not get any pest attack. That kind of bacteria, that gene is added to this plant. Sir, it is a good thing, right? It's a good thing. Instead of using pest, we have developed a plant that is pest resistant. See, in case in future, have you heard the name Odomas to get rid of mosquitoes? If we get the gene which produces Odomas naturally, or skin producing Odomas with sweat, in case we will not be bitten by mosquito. That kind of thing happening here. It is producing a chemical which is pest resistant, which resist pest, BT brinjal, BT cotton, right? That can be useful in many terms, but there is a problem. What problem? It will affect the food chain, food web. If there are no insects, the animal organism dependent on that insect will die. The food chain will be affected. That in turn will affect the farmer himself, right? The food chain will be affected, and there are so many side effects by having this genetically modified crop food product. Many side effects like allergy, etc., can happen. That is the reason the usage, it was restricted. It got banned in India due to various farmer protests. In the long run, in the shorter period, it will help the farmer. No need of pest control chemicals. But in the longer run, it will affect the biodiversity, the food chain. That's why it got banned. What we need to know, BT cotton means not biotechnology cotton. BT cotton means Bacillus thuringiensis. Right? That's it. Implementation of biosafety guidelines by Department of Biotechnology, then establishing an information network throughout India. We are doing so and so research. If you want any information, you can get that kind of network nationally, internationally, and between the scientific community they are doing. National Biotechnology Board, established in 82. No need to remember the year. National Biotechnology Board. Their main function, strengthening the indigenous capabilities to develop new technology, to develop new biotechnology products. That's it. Department of Biotechnology, established in 86. Department of Biotechnology works under which ministry? Technology. Ministry of Science and Technology. Helps in the development, innovation, manufacturing in the field of biotechnology. Department of Biotechnology. Then we have National Center for Microbial Resources. National Center for Microbial Resources. We have vast, wide variety of microorganisms around us. See, if you have the habit of reading newspaper regularly, you might have come across recently in our Western Ghat, a new frog have been discovered, a new insect have been discovered. This kind of news you might have seen, right? That new organism, actually that organism is not new. It is new to us. We have discovered it for the very first time. We have named it with some scientific name, right? Even that small organism has a greater impact on the biodiversity. Let's say, for example, from tomorrow, there will be, if in case no honeybees in the earth, what happens? Zero honeybees in the earth. Pollination stops. Seed will not form. We will not get food. The biodiversity, it will collapse. Many organisms will die in the shorter period. Longer period, it will affect us also. Very bigger impact it is having. The honeybees, we feel it is a small organism, right? Likewise, there are so many microorganisms which we have in India. We need to protect them. We need to have their data. For that, we have this organization, National Center for Microbial Resource, the diversity to know, conserve it. Then Indian Biological Data Center, India's first national repository. Repository means kadata, to store the data, ledger kind of thing, right? For life sciences data means what kind of animals, organisms we have, etc. All that data. Government of India initiatives for biotechnology in India. One is National Biopharma Mission. See, listen, for the UPSC examination point of view, if there is any mission that has been introduced in this year, that will be asked in the preliminary or mains examination. If it is not introduced, established this year, if the older mission is in use due to some any updation or implementation, then also it might be asked in the examination, right? Then in the case of mains examination, they might ask, please shed some light on the development of biotechnology in India. That means I don't know anything. I'm asking you, please tell me how biotechnology, the field developed in India. Then you have to say, once upon a time in 1986, the Department of Biotechnology started under Ministry of Science and Technology. Then we have so and so organizations which do this job, right? You need to explain, right? To write the mains examination, you need all this data. For the prelims point of view, you should know trending recent organization or committee which is in news. In that committee, you don't have to know the name only. You need to know the details, what it is doing, how much money it is spending, etc. You need to know the statements they will give and ask the question, right? National Biopharma Mission, collaborative mission between industry and academia, means institution, colleges and industry, to accelerate a biopharmaceutical development, medicine development in the country. Then I3 program, Innovate in India program, enabling ecosystem facility infrastructure to help entrepreneurs indigenously manufacture many biotechnology products, helping biotechnology startups. That's it, I3. This will be implemented by a special council called Biotechnology Industry Research Assistance Council. Biotechnology Industry Assistance Council. Then Biotech Kisan, launched in 2017. See, the main problem with our farmers and the government is introducing many schemes, but we are failing to reach those schemes to the farmers, right? Let's say your father, grandfather, your relative, he's a farmer. Do you know any farmer who is in regular contact with your local Krishi Kendra? No. Do that farmer know any of the schemes? Generally, he will be knowing the schemes where he can get some money by making some Krishi Honda, etc. That kind of schemes, right? Other than that, we have many schemes. Basic free soil testing is done. Free soil testing. If I purchase a land, I should first do the soil test. What are the minerals that are less in my land? What I need to add? Which kind of crops I can grow in this land based on the amount of water which I have right now, right? And then By visiting the agricultural department, I will get the idea how much area of my district or state is being sown with what crop will be financially beneficial for me to grow this year. See, each and every person, by listening to someone grows the same crop tomato, the rate will be 1 rupee, 2 rupee kg. Later, everyone stops growing tomato, then the price will be 80 rupees, 100 rupees kg. That is the fluctuation which we are seeing from generations to, right? That is happening. We need this collaboration between the department and the farmers. This one, Biotech Kisan Mission, connecting scientists with farmers, science laboratories with farmers. If a person do agriculture by inculcating technology, biotechnology, 100% he will be financially stable, better. He can earn good money. That is the main problem which is happening in our country. Atal Jai Anusandhan Biotech Mission, implemented by Department of Biotechnology. I'll share the PDF. To address the challenges of maternal and child health. See, after giving birth, the mother requires various nutrients, right? To address that deficiency in the mother and children, this mission to develop various nutritious food alternatives for the natural food which the mother is getting, he will get a nutritious alternative. If you go to Anganwadi, you will get food. Generally, we say food, right? That is a nutritious alternative, a mixture which we can add to water, milk, etc. Right? That kind of thing. With the help of this mission, to study antimicrobial resistance in mother, child, and to study the effect of vaccines and to develop new vaccines against the new diseases which we are getting, then food and nutrition, etc. All this comes under Atal Jai Anusandhan Biotech Mission. One Health Consortium, Department of Biotechnology, established in 2021 to survey important bacterial, viral, and parasitic infections, important disease causing microorganisms. To study that, One Health Consortium, BIRAC, Biotechnology Industry Research Assistance Council, to help the entrepreneurs, new companies. Biotechnology Park, Biotech Park. Tech Park means what? In Bangalore, we have many tech parks, right? Each and every facility required for the company will be their infrastructure. The company just needs to get in, take a space, and start its business, right? Tech park. Furniture will be there. Meeting hall will be there. Each and everything will be there. You need to just pay the rent and work there. Tech park. That's it. In the tech park, many number of companies will be operating. Right. Likewise, biotechnology park means government will provide all the facility required, laboratory assistant, etc. The companies, the new company, startup will start business there. That kind of thing. Like the industry parks. Startup landscape. India is planning to increase the number of startups by 10,000. Earlier, it was only around 50. Few years, 10 years ago, it has been increased to 6,500. We are planning to have 10,000 plus. Actually, it is around 8, 9, 10,000. It will be increased in the near future. Genetic Engineering Appraisal Committee. Works under Ministry of Environment, Forest and Climate Change, not under Department of Biotechnology. Genetic Engineering appraisal committee. This deals with if you use some pesticide, if you use some medicine, how will that affect on the environment? That will be studied with the help of this organization, right? Large scale use of hazardous microorganisms, then recombinant technology and industrial production. How will that affect the environment? That will be studied. See, for example, in the earlier days, during the time of plowing season, behind the plowing tractor or the farmer, there will be many birds trying to eat the worms coming out from the land, right? These days, no worm in the land. Why? We used many pesticides that will in turn affect the food chain. That will be studied with the help of this committee. Comes under, works under Ministry of Environment, Forest and Climate Change, right? Genetic Engineering Appraisal Committee. Wheat genome sequencing program, rice functional genomics, crop biofortification. Genome sequencing means possible genetic sequence which is available in India. We have many breed types of wheat, right? We have many types of rice at least, right? Basmati, Namasuri, Rajamudi, etc. varieties we have, right? Each and every rice will be having different genetics. Different genetics. That data will be collected. Rice genomic project, wheat genomic project. If you have that data, let's say for example, you have 4 members in your family. Okay. One is having travel sickness, motion sickness. One is having allergic to dust. One is having an issue with the air conditioner, AC. If you know them better, you can handle them better, right? A person is having travel sickness, you avoid taking him to trips. A person is having issue with the dust, you will keep him out while cleaning your home. You can avoid, right? By knowing the genome sequence, we can we can know how a medicine, how any product which will how it will affect on the plant, wheat, etc. Like that. Let's say for example, I will introduce new fertilizer, I will introduce new pesticide, I will introduce new herbicide. How will it affect the present wheat, present rice? Rice in the sense paddy. That's what.